Okay, so we wrapped up with transcription. So we've already made the RNA copy of DNA. We transcribed that gene, we edited it if it was an eukaryotic organism. So now that messenger RNA, after it's been edited, we call that piece of RNA a messenger RNA or mRNA. Now that mRNA is going to leave the nucleus, it's going to exit that nucleus, and it's going to find a ribosome. These ribosomes can be free-floating, they can be hanging on with a rough ER. Regardless, it's going to find a ribosome, and it's going to then start the process of translation. So that's our next step in gene expression, of getting that information in the DNA, in the gene, into a protein product. DNA to RNA is transcription. So RNA to protein is translation. So we're going to translate that nucleotide language into the language of amino acids of proteins. So we have some new molecules in here that we need to talk about first. Okay, so we're going to introduce more in depth what a ribosome is, tRNA, and a codon. Okay, so these ribosomes, we've talked about them before as far as that they're the protein factories. But what ribosomes are two pieces of what's called ribosomal RNA. So the ribosomes themselves are actually built out of RNA, and their main job is they're going to be creating a platform for the RNA transcript to sit on, and then it's going to catalyze the connection of those amino acids. Okay, so amino acids, those are the monomers, the building blocks of proteins, they're going to be connected by the ribosome acting as a catalyst to bond them together. But we got to get the amino acids to the ribosome, and that's where this next molecule, tRNA, or transfer RNA, the T stands for transfer. Transfer RNA is another molecule built out of RNA, out of a specific type of RNA called tRNA. And it's going to be carrying amino acids. So tRNAs, we have 20 amino acids. tRNAs are just floating around the cytoplasm, and each tRNA has a specific amino acid attached to it. Okay, so they're hanging out, they're floating around, and they're going to be brought into the ribosome with that amino acid. And the way that tRNA knows which amino acid to connect to is it has these three bases right here. So those three little like teeth-like thing that are sticking down, those are what we call an anticodon. So those are nucleotide bases, A, U, C's, and G's. There's no T in RNA, so it's A, U, C, or G, a three-letter sequence. This anticodon on the tRNA is going to be complementary to the codon on the RNA, on that messenger RNA. So if there's an A in the messenger RNA, we would find a U in the tRNA. Okay. And so the DNA sequence is going to be read in sets of threes. And those three letters, that triplet code, that three-letter sequence, we call it a codon. So every three letters of the sequence that's in DNA and then that gets transcribed into RNA is going to code for the information for one amino acid. So every three DNA and RNA bases codes for one amino acid. And it's the codon that's on DNA and mRNA, and the anticodon on tRNA is complementary to the codon on the mRNA. Okay, and that's how that amino acid is going to match up with that mRNA. So that anticodon on the tRNA and the codon on the RNA. So how is this read? So we are going to be using a chart like this in class to read the genetic code. And this is really cool. We've cracked the genetic code. We know which codons code for which proteins. So the way we read this, so let's say we have a transcript that has a codon that is ACG. ACG, that's our codon, ACG, on the RNA, on the messenger RNA, ACG. So what we would figure out, what amino acid that would code for, is our first letter, A, is right here. Okay, our second letter was a C. So you're going to move across this row until we hit C. Okay, so here's the C column right here. And then our third letter in our codon was a G. So ACG, that codes for the amino acid threonine, or THR. So each of these three letters in here, the proline, serine, SER, PHE, these are all shorthands or abbreviations for that amino acid. But a couple things that we want to notice here, there's some of these that are in red, these ones up here. and this one right down here is in green. So this green codon right here, AUG, we call this the start codon. So AUG is the start codon. What that means is that all RNA transcripts are going to begin with that AUG codon. So that means that all proteins, when they're being constructed, that first amino acid that's going to be connected is going to be methionine. So AUG is our starting codon. So all messenger RNAs have that first codon AUG. That's where the beginning of translation occurs. And then the rest of the codons in that transcript are going to be any combination of any of these other amino acids. And then the way that the ribosome knows to stop translation is if it hits one of these stop codons. So these ones in red, these are the stop codons. So UAA, UAG, and UGA, those are 3 codons that do not have a matching tRNA. There is no complementary anticodon in the tRNA that will match up with those stop codons. And so at that point, the RNA would dissociate from the ribosome, and then translation would stop. So these stop codons indicate that this is the end of the process of translation. Okay, and we'll practice this a little bit later on. So how this works is, first thing that's going to happen, that mRNA, that messenger RNA, is going to associate with the smaller piece of that ribosome. So I kind of think like ribosomes look like weird hamburger buns. So we're going to have the lower bun of that ribosome, the smaller subunit of that ribosome, is going to connect to that RNA. The RNA is going to sit right on top of it. And then our first tRNA that's carrying that start amino acid, that methionine, that AUG codon, that start codon, that methionine carrying tRNA is going to land, boop, and that anticodon on the tRNA is going to match up with the codon of that start codon on the mRNA. Okay, so that's how everything starts. So once those pieces are in there, the lower bun, the smaller ribosomal subunit, the mRNA sitting right on top of it, and then the methionine tRNA. Okay, so that's how we begin translation. Once all those pieces are set up, the larger ribosomal subunit, the top bun, if you will, is going to come in. It's going to connect as well. Okay, so this larger ribosomal subunit is going to come in, and what this is really doing, this is where the catalysis happens. So this larger ribosome subunit, what it's going to be doing is, as new tRNAs come in that are charged up with their amino acid, so this anticodon right here, AAA, this matches up with the codon UUU on this piece of mRNA, and that is the anticodon that's carrying phenylalanine, PHE right there. Okay, so what the larger ribosomal subunit does is it takes those 2 amino acids and it forces them to bond, kind of like an enzyme. It's a catalyst. So it's going to catalyze the connection of those 2 amino acids together. It's going to create a peptide bond. So it's the bond between amino acids. We call that a peptide bond. And so once it starts to connect those amino acids, it creates a polypeptide chain. And so that polypeptide chain is the first step in the building of that protein. So it's going to connect those 2 amino acids together. So the methionine gets connected to this phenylalanine right here. And it's going to be attached onto that phenylalanine. It's going to grow out of that phenylalanine, just like we see here. Then it's going to move over. It's going to move over. This original tRNA that was carrying methionine, it exits, it leaves, it's going to go get charged up with another methionine. Then once it moves over one codon, it exposes the next codon right here. Okay, so the CGA codon, it's going to match up with the anticodon GCU. And this tRNA, it's carrying that anticodon GCU, is carrying the arginine amino acid. And guess what? It's going to have the same thing happen. That phenylalanine right here, the ribosome is going to catalyze the bond between that phenylalanine and that arginine. It's going to create a peptide bond between those 2 amino acids. And now our polypeptide is going to be 3 amino acids long. Okay? And so it's going to keep doing this process of connecting these amino acids and moving over one codon, connecting, moving, until it hits one of those stop codons. And then everything dissociates. That polypeptide chain floats away. It's going to get folded up into its final protein shape, maybe connected with some other proteins. And then that mRNA reused, those tRNAs get charged back up again, and then that protein could get made again, or that ribosome could find another mRNA and make a different protein. Okay, so all these pieces can be recycled. The mRNAs will get degraded over a certain period of time, depending on the organism. It can be 20 minutes, it can be a couple weeks, it varies. So those mRNAs can be reused, or they can be degraded. The tRNAs are recharged up, the ribosome gets reused, and it can go through this process again and again. Okay, so let's practice. So let's say we have a DNA sequence right here. I want you to pause the video real quick, and I want you to see what is the complementary DNA sequence. I want you to write that down. Don't forget your 3' and your 5' ends. How would you transcribe this DNA sequence, transcribe this DNA sequence right here up top into an mRNA? Okay, so transcribe this DNA into an mRNA, and then that mRNA that you create, translate that into protein using this chart right here. Okay, so go ahead and pause. Alright, so hopefully you paused and got all of that done. So here we go. So if we were to find the complementary sequence of this DNA strand, here's what it would be right here. So A matched with T, G's matched with C. Okay, and notice the 5' and the 3' end. So in this end we have the 3', because DNA is anti-parallel. We would find the 5' end over here. So they're running opposite directions from each other. Okay. Now if we were to transcribe this DNA sequence up here into RNA, here's what we would have. And I broke this up into 3s. This is going to be helpful for you also, is as you're transcribing, break it up into 3. So these dashes just indicate separations between codons. So UAC, CGU, this is what happened when we convert this DNA strand, when we transcribe this DNA strand into RNA. Notice again, here's our 5' end, because it's going to also be anti-parallel to our original DNA strand, which is running on 3' on this end. Okay. So now if we translate this into protein, into amino acid, our first codon UAC, so let's find that. So here we go. So here's our first letter on this side, U. Go over to our second letter, A, and then our third letter, C. So this would be the amino acid tyrosine, TYR. Okay. Our second codon, CGU. Our first letters in that codon is a C. Our second letter is a G. And our third letter is a U. So arginine, ARG right there. Okay, so that'd be our second amino acid. ACG right here, this codon. A right here is our first letter. Go over to C. And our third letter is a G, ACG. THR, that's threonine. Okay, so there's our next amino acid. GGC is our next codon. So G is our first letter. G is also our second letter, and then C is our third letter. So GGC right here is the amino acid glycine. Okay, so GLY. And then we have also UAC again. UAC, we had that at the beginning, that's tyrosine. So UAC. All right, and so that's how we transcribe and translate a gene into its final protein product. This is how genes are expressed. This is how we turn genes in our bodies into things like our proteins, like our hair, our fingernails, our enzymes in our body, everything. Transcription, DNA and RNA, and then translation, RNA to protein at the ribosomes. All right, good job.